Yes, we just sort of finish off the last four slides from our discussion, project them and discuss them.
Okay, so I'm just going to finish up the last 4 slides on the cell cycle lecture just to complete that. So that probably takes about 15 minutes, and then Dr. Danielowski is going to come in and he's going to start his content. He's going to make some announcements about just sort of course-related issues. He's also going to talk about tests. As I'm sure many of you are aware, there were some interesting things that happened yesterday. We are fully aware of it, we're not stupid, and we're going to have to address that. It's also an issue, it's also interesting to see what people's morals are. And I'll leave it at that. Let's just say I'm very displeased with what happened, and we will have to correct for this. Okay, so we're going to finish up talking about the cell cycle and its regulation. Last lecture we talked about the anaphase promoting complex, which is an E3 ubiquitin ligase, which is responsible for turning off mitosis by degrading mitotic cyclins. What I didn't talk about was how do you get rid of cyclins at different phases of the cell cycle. So when we look at, for example, the association, for example, if we want to start the events to start off entry into the S phase, we're first going to have events happening in G1. So G1 S phase cyclins will bind to CDKs and cause expression of genes, some of which will encode the S phase cyclin. But we don't want the S phase cyclin to be active right away. So there's an inhibitor protein that binds to it. So this is happening in G1. So you start to get increased transcription of S phase cyclins in G1, and to prevent the premature activation of CDKs, there's a protein. It has a name, and I don't want to give any more names we already have in this course. So it's this inhibitor protein that binds to this complex, and it prevents S phase cyclin CDK complex from being active too early. But as you progress further into the later phases of G1 S phase cyclins can be active, and what they do is phosphorylate that inhibitor protein. That in turn acts as a signal for a protein called SCF. So that's a E3 ubiquitin ligase, and what it does is actually promote the degradation of the phosphorylated inhibitor protein. So it promotes ubiquitination, that leads to the degradation of that inhibitor protein, and now the S phase cyclin CDK complexes are activated, and they can turn on a number of genes that are involved, for example, in DNA replication. So SCF kind of has a very versatile E3 ubiquitinase, so it kind of does multiple duties. So this is the first stage here, is degrading an inhibitor protein to activate this complex. But it will then go on at certain times, it gets activated through a whole bunch of real complex mechanisms, which I'm not going to test you on, that it can be activated at later phases of the cell cycle to degrade other cyclins. So to put it all into context, this is sort of the whole big picture here. As we start off in G1, we're going to have G1 S phase cyclins. They are going to promote the expression of S phase cyclin, and that will start to associate, the S phase cyclin will associate with CDK, but we don't want that to be turned on right away. So there's an inhibitor protein that's present that will bind and prevent its premature activation in the G1 phase. As we progress towards the end of G1, the G1 S-phase CDK complexes, the kinase activity is turned on. It will phosphorylate the inhibitor protein, and that in turn will make it recognizable by the SCF ligase, which will promote its degradation. So now the S-phase CDK complexes can trigger increased expression of DNA synthesis, and the SCF will also go back, and it's not shown here, degrade G1 phase cyclins. As we progress throughout the S phase towards the end, we now need to turn off this complex, and the SCF, through a complex mechanism which I'm not going to test you on, will promote the degradation of the S phase cyclins. So SCF plays a major role in controlling cyclin levels, CDK activity, at G1 and S phases. So when we're looking at ubiquitin, E3 ubiquitin ligases, SCF has kind of multiple roles at both the G1 and S phase. Now, as we're moving forward into G2, we're having the accumulation of mitotic cyclin, which is associated with CDK. And to prevent its premature activation either in S or early G2 phase, the V1 kinase will phosphorylate the tyrosine at position 15. That's an inhibitory phosphorylation. So that prevents any premature activation of the MPF complex. As we move forward into G2, as we get towards the end, now we need to remove that inhibitory phosphate group. That's achieved by the CDC25 phosphatase that takes off the inhibitory phosphate group. And we have activation of that kinase CAC, which will put on the activating phosphate group at threonine 161. So now we have a fully active mitosis promoting factor complex that's going to phosphorylate things in terms of promote nuclear membrane breakdown, mitotic spindle apparatus assembly. And as we progress through to anaphase, where the chromosomes are now starting to be pulled apart, the cell has to prepare for forming two new cells, two independent nuclei, and so we need to shut off the MPF. So that will be achieved by the anaphase promoting complex. That's the other E3 ubiquitin ligase, which will promote the degradation of the mitotic cyclin shown up there. And then that will allow anaphase to progress into telophase, and ultimately the cells divide into two cells. So if you can go through all of these stages and say close your notes, and if you had to draw this all out and all the details of all the players and all the stages, that's what you need to know for the cell cycle. So we spoke, the first part of the lecture, we talked a lot about this, and then now I've sort of focused, filled in some blanks here. So I mentioned that sometimes cells can exit the cell cycle and go into G0. Now, but quite frequently that happens when there's some kind of unfavorable, there's not enough nutrients, not appropriate signals to trigger replication of cells, cell division. So that's what we refer to as restriction point. So kind of everything goes along, and there's a sensor, just call it a sensor, that determines whether the environment is favorable for the cell to divide. And if it isn't, the cell will exit G1 and stay into G0. There are other things that can happen that we don't want the cell to replicate its DNA. And the most important aspect is DNA damage. We don't want DNA damage to be replicated because that could be, for example, a mutation in a really key gene that might regulate cell cycle or apoptosis. And so we don't want that mutation to be propagated. So usually the cell will sense the DNA damage and could trigger activation of DNA repair enzymes, which can then repair the damage, or if there's excessive DNA damage, can actually trigger apoptosis. So there's a number of these DNA damage checkpoints in both G1, S, and the G2 phase. So obviously damage initially in G1, we don't want to enter into S phase, so we need to deal with it there. Or at the end of S phase, when DNA is being replicated, it has to unwind a little bit. It's very susceptible to damage at that point. And so after there's been replication of DNA, there's also sensors to determine has it completely replicated the DNA, and has it repaired any damage that might have occurred during DNA replication, or errors that occurred during DNA replication. And then entering into G2, again, is there any DNA damage that's happening in there? And then finally, mitosis is important to ensure that the chromosomes are properly attached to the mitotic spindle and separate appropriate. So at every stage here, there's a number of things to make sure that the genetic information is preserved, it's accurate. And that comes at the level of the DNA, and also that the chromosomes are equally segregated. And so there's various sensors that monitor this. I can't go into every one. I'll just focus on this one, which is the P53 retinoblastoma system. So many people have probably heard of P53. It's mutated in a lot of cancers. If it's not functional, it can allow DNA damage to be propagated, and that can lead to a cascade of events that lead to uncontrolled cell growth in terms of DNA damage that affects a lot of regulation of the cell cycle itself. So there's a number of DNA damage sensing proteins, and I'm not going to name them because it's not really that important. Just know that quite frequently these DNA damage recognition proteins, either themselves or they can trigger activation of the protein kinase. And the kinases can phosphorylate P53. And phosphorylation of P53, depending on which phosphate group, which recipe, is generally associated with the activation of P53. Now normally, P53 is made all the time, but it's degraded because it is recognized by MDM2. It's an E3 ubiquitin ligase, which promotes the ubiquitination and the degradation of P53. When it's phosphorylated, it doesn't interact as well with MDM, and phosphorylation of MDM itself, MDM2, inhibits its activity. So as a result, we have now the stabilization of P53. It's no longer being degraded by MDM2. It will accumulate, enter into the nucleus, and either directly act as a transcription factor or affect other transcription factors. In this case, it can increase expression. It increases expression of many genes. This is by no means, it's not a one-trick pony. It does many things. But for the context of this course, we'll say that one of the genes that it turns on is a gene called P21. So P21 is a cyclin-dependent protein kinase inhibitor. And what it will do is bind to CDK cyclin complexes. So I'm not going to get into, wait a moment, we never talked about CDK2 or 4 or cyclin D1 or whatever. It's not important. As I mentioned, yeast have one CDK, mammals have many. And there's more complexity in terms of mammals have a number of different cyclins. So these particular cyclins would be like G1S cyclins. And so it's not really important to remember what D1 and E is. It's just important to know that cyclin CDK complexes that are associated with G1S phase transition will be bound to by P21. And when P21 binds to these complexes, it inhibits their kinase activity. So when their kinase activity is inhibited, that will actually lead to cell arresting. Why? Because under normal circumstances, when these complexes are active, they will phosphorylate this protein called retinoblastoma 1. And when it's phosphorylated, it can no longer bind to this transcription factor called E2F. So there's phosphorylated retinoblastoma 1. It's released from the transcription factor E2F. That E2F can turn on things related to DNA, chromosome replication, and also increased expression of cyclin genes. So this is a major transcription factor which turns on those things that help promote the transition from G1 to S. And so when we have DNA damage, activation of P53 turns on P21 expression. The P21 protein will bind to and inhibit this complex, and therefore it's going to prevent the phosphorylation of retinoblastoma 1. Unphosphorylated retinoblastoma 1 binds very tightly to the E2F transcription factor, and it prevents its transcriptional activity. So it can't turn on those genes that will promote DNA replication and chromosome replication. So at this point, what happens is the cells essentially arrest in G1 because they don't have the cues to progress further. And at this point, there can either be repair of the mutations, or P53 can turn on other genes that can trigger apoptosis. So collectively, mutations in either P53 or retinoblastoma are associated with cancer. Just the mutations in those cells can make the cell very susceptible to cancer. Why? Because DNA damage is basically going unchecked. And if there's enough DNA damage, eventually it's going to fall into really key regulatory type proteins, which we'll talk about towards the end of the course, that can lead to uncontrolled mitogenic signaling, or the inability to shut off the cell cycle progression at appropriate points. So again, when we have DNA damage, you're going to get an increase in P53, an increase in P21, and that is going to, by inhibiting this cyclin CDK complex, prevent the phosphorylation of retinoblastoma protein. It's going to be maintained in that state, and we're not going to allow E2F to go on and activate gene expression. So that's pretty well my content for now. Dr. Damianowski is probably going to come in the next minute. I told him to come a quarter to, so a minute ahead of time. He's going to get into a whole bunch of other really interesting stuff for 12 lectures. And then in the final 4 lectures, I'm going to come back. We're going to talk about signal transduction mechanisms. So we'll have a very brief pause until Dr. Damianowski arrives. So I've been using that. It's kind of a confusing, so you gotta try to work on. Don't turn it on and it works. You just press it once and it comes on, and just turn it on and it should happen as fast. So somebody since last week dropped it because it didn't have a big dent in it. Okay. You got your own cord? I got my own cord. Thank you. So I already kind of forewarned them that we know what they're up to. We'll talk about morality as well. Lack of.
Oh boy. Try not to yell at them. They know what you're doing.
All right. So
I'll
good?
see you this afternoon. Yeah, I think so. No problem. Yep.
All right. Have fun.
Yep.
I shall.
All right, hello. I'm going to try with the mic on my tie. Hopefully this will work. If you can't hear me, please do something, wave or whatever. So I am Dr. Sashko Damianowski. Hopefully you guys remember me. We met at the Seats a few weeks ago. No. I introduced myself when the course first started, and I'm going to be with you for the next 6 weeks, giving you 12 lectures. So I'm going to remind you of a bunch of things, yell at you guys for a little bit, and then we'll get on with the lecture material. All right? So the topics I'm going to cover are sort of related to the cytoskeleton and cell adhesion. So there are no required readings. The material that you're going to be tested on is on the PowerPoint slides. However, the material is in these parts of the book. Right, so this is the page numbers and or section numbers in the text where the material is found. So if you want to read ahead or whatever, this is where it's found. This should be available to you. Okay, so these are the sections, and this is where in the textbook things are found. Okay, so it's going to be for all of my lectures. Kind of remind you what's happening in the next little bit. I'm here for 12 lectures. So Dr. Cumming is going to give 12 lectures. I'm going to give 12 lectures. The way it worked is he gave 8, I'm going to give you 12, and then he's going to come back and give you the final 4. Okay? So you had 4 lectures, a test, 4 lectures, a test. I'm going to have 4 lectures, then there'll be a test coming up on February 14th. Then I'll give you 4 more lectures, and then there'll be another test on a Sunday. So there's going to be basically 4 lectures, a test every 4 lectures. After that, after the tests, I will have 4 lectures, and then Dr. Cumming will have 4 lectures. So the last 8 lectures are on the final exam. Okay? So there's a test coming up on February 14th, which is a Friday, Valentine's Day, but love stinks, so it's okay. So, February 14th is the day before reading week starts. In principle, we can't have evaluations on the weekends because that's part of reading week, so that's why this midterm is on February 14th. Okay? The final exam is going to be in April, set by the registrar. The final exam is cumulative. The tests will not be cumulative. So my first test will be on my first 4 lectures, my next test will be on my next 4 lectures. Okay? That part makes sense? Like Dr. Cumming, I have office hours, so that'll be 2:30 on Mondays and Wednesdays upstairs in my office in BGS 3053. So I'm available then. I am available outside of that time if you can't make it because of the classes. You can send me an email and say, hey, can I come by Thursday at 10 a.m. or whatever, and I'll tell you yes or no. The OWL forums are pretty well used so far. Use the OWL forums. If you guys have questions about course materials, Libby and Brad, the TAs, are on there answering questions, and we look at things to make sure that that you guys get things. And it's nice to kind of see how you guys are thinking. Email me questions is not reasonable. Trying to send me an email, sort of, saying, can you please explain lecture 3 to me? It's impossible to do that over email. Okay, so if you have questions about course material, either use the forum or come up during office hours or email, and see if you can cut it at a different time. The tutorials are almost up. They're kind of up, and we'll talk about them in a second. They will also have office hours. Josh Pemberton will have office hours, probably over Zoom, probably on Fridays, but you'll get more information about that in the next day or so. All right, test 2. That's finished yesterday. A number of times over the course, I'm going to tell you how much I hate Brightspace because Brightspace is awful, and add Achieve to that, which is the other software. It just makes things awful. So the definitions between end dates and due dates and releasing feedbacks and publishing and releasing grades, it's not intuitive. So in the software, it's very difficult to try to get stuff out to you guys, to know when you guys will see what you will see. Yesterday with the first test that we had, we didn't allow you to see anything, and it took us several days to release your answers. Apparently, excuse me, you can set it up so that answers will be released the next day. So it was set up that answers would be released today at 5 p.m. Except they weren't, of course. Answers were released when people completed things, despite the setting that should be released today at 5 p.m. So answers were out there, and apparently, because you guys had no scruples or morality, you guys were sharing answers with people. So, that is the grade distribution. It's kind of funny, but we got to do something about it. All right, so there's a clear pattern where people who wrote the test early didn't do as well as people who wrote the test later. Okay, even though we figured out what was going on and we shut off all the answers, we shut off everything, your grades are no longer even visible in test 2. But clearly answers were out there, and people just felt compelled to use them even though you know it's wrong. You know cheating, you can get expelled. You paid lots of money, don't do it. Okay? You guys, everything you do is logged in. We know when you logged in, we know when you answered questions, we know when you logged out. Many of the people who scored 100% did so in taking the test between 8 and 12 minutes. That's pretty damn good. We know who did that. Do we prove that you cheated? No. But come on. Okay? Don't do that. Be fair. You're not going to get away with things. The final exam is going to be in person. You got to get used to the material. You got to get used to answering questions, studying, and doing things. What are we going to do about this? 2 or 3 things. One is somehow bell up the marks of the people who did things early. Not sure exactly how to do that, but we will try to adjust people who wrote early, who didn't have the answers, or other sort of manipulation. We will bell something upwards. We'll see how that goes. Not sure exactly how that's going to work. Number 2 is likely because you guys are shooting yourself in the foot. This 10-hour window that allows you guys room to cheat obviously doesn't work. So for the next midterms, you're going to have a smaller window, possibly a 2-hour window, or even just everyone writes in that 1-hour window or something. You did it yourselves. Okay, so there's going to be a lot less room for you guys to do things, and you will not get your anything released until days afterwards. All right, we want you to do well. We are thrilled when students get good grades, but this is not reasonable. Those questions were pretty tough. Some of those questions, just reading them took you 3-4 minutes, never mind answering all 20 questions in 8 minutes. And you're getting 100%. Okay, so it'll be a couple days before we figure out how to adjust things and manipulate things. And how the test 3 is going to go along, I don't know. I know my daughter always tells me I yell too much. I'm not yelling at you. I'm not disappointed at you. I'm just saying, behave, please. Make my life easier. I don't want to have to do stats and bell curves. I got other things to do. All right, so you'll get your mark at some point. Some belling will be done. How will we move forward? We're not doing anything about this. We can't. We're not going to remove the grades. We're not going to remove the test or anything, but it's unfortunate. All right. Moving on to Achieve. Achieve is a software module that's available from the publisher. So this course is a joint course between Schulich and Faculty of Science, so Faculty of Medicine and Faculty of Science. And for decades, we've been trying to get a tutorial back into this course. In principle, courses should have more than just lectures. You should have some sort of other component. So a few things happened where suddenly that seemed kind of feasible. One, we had a new faculty member hired, Josh Pemberton. 2 was that Brightspace was being implemented. So Brightspace is sort of a more modern software. And with Brightspace, we can then use Achieve. So Achieve is this software that the publisher provides, where in principle, there are all kinds of activities in Achieve that can be used for tutorials. So the publisher, met with the publisher many times, and meeting with the publisher is going on a first date. They're polite and wonderful, and they treat you nice, and they promise you things. And then after you commit, it's like totally different. So they had guaranteed there would be seamless integration of Achieve into Brightspace. There's nothing seamless about integrating anything from Achieve into Brightspace. It's horrible. So what we wanted to do in Achieve and make it in Brightspace doesn't work. So we've been struggling how to do things. And it's finally coming together where the tutorials are just videos that you can watch the videos. Those videos will be in Brightspace. So in Brightspace is one video at the moment. There'll be another one maybe this afternoon, and those are the first 2 tutorials. So you watch tutorials, and then you will answer a quiz. The quiz is in Achieve. That means everyone needs to buy Achieve. Okay, I apologize. I know I'm going, no. It's got to be done. We're committed. We already shook hands with the devil, sort of thing. Right? So the quizzes, the tutorial quizzes are in Achieve. They are linked through Brightspace. There's a link in Brightspace, but you need to buy Achieve to do the tutorial quizzes. Achieve has many, many activities that are useful for the student to learn not only the tutorial material, but all the lecture material. Interestingly, when you buy Achieve, you don't get access to any of that, which is ridiculous. We have to give you access to all these things in Achieve, and it's not clear how things are organized. So in the past, or in the last bunch of lectures, there's been no Achieve resources in Achieve. Now there is. We're starting to put stuff in there. There's movies and little exercises and a whole bunch of other stuff that's present in Achieve that are now finally becoming obvious because we're putting them in for you. As opposed to, since you paid for it, you should have got everything, but that's not the way it works for some reason. So in the Achieve website, under My Lectures, there are a bunch of exercises and fun things to do and games and readings and stuff. Hopefully they're useful. They are not testable. Okay, so in Achieve, there'll be lots of things for you to look at. They are not testable. All the test stuff comes from the PowerPoint presentation. Right? The tutorials, you tested on the stuff in the videos. Okay? The first two videos are incredibly long. Sorry. Okay, the first video that's up is long, it's interesting. The second video that's going to go up is going to be even longer. There's only 10 questions from the two videos. Achieve quizzes, the way they do their quizzes, even though they said it's all powerful and you can do whatever you want, you can't. So all the questions are going to be visible to you at once. So you will have the 10 questions sitting there, and as you watch the video, you have this 10 questions there, so you can see the 10 questions, you're watching the video, you can kind of get the questions as you're watching the video. There is no time limit, the quiz is available for basically a month. And you can write it 5 times. Okay, that sounds good. The part that isn't good is that when you submit, you will get how many you got wrong, but you won't know what you got wrong. Right? So there is no way to tell you what you got wrong. You will get 7 out of 10, or 8 out of 10, or 6 out of 10, but you don't know what you got right, what you got wrong. So it's up to you to figure out some sort of strategy without cheating and using all your friends to do that, please. Make sense? Okay, so there's going to be 3 Achieve quizzes, which are 2 tutorial quizzes, and the top 2 count. For all of them, they're going to be available for a long period of time. It's up now, the quiz is up now, except the second tutorial video isn't up yet. Josh, Dr. Pemberton will have Zoom office hours, hopefully starting this Friday, but we'll post more information once we post up that second video. The second video, because it's long, might be in part A and part B. What else we got here? So again, there's lots of resources in Achieve for the lectures. The lecture Achieve resources are not testable, they're just fun, and hopefully you guys will enjoy them. There's also other little videos that are in Brightspace. So we had little activities over the years for you folks, and we thought we could put them into Achieve, but you can't, of course. So there's little activities, learning activities with my lecture. You can just kind of play around hitting plus signs or dragging and dropping names or whatever, fill in the blank. There's a bunch of them coming. But again, those are just for your entertainment and not so much, they're not testable. Phew. Right. We good? Questions with all that? People want to run screaming? Okay, get that up behind us. Let's all be happy now. Okay, and start off with my lectures, which are going to begin with the cytoskeleton. Okay, I'm going to go slow, sort of an overview and an introduction to things. I'll try to explain what I put on slides and what you guys should get out of things. A lot of times I just kind of talk over introductory slides, which are sort of these first 2 slides. There's nothing really to memorize, just kind of sit and listen about things, all right? So we're going to be talking about something called the cytoskeleton and cell adhesion, right? So why is that important? Well, we're multicellular organisms. We're made up of trillions of cells. So these cells have to be held together. So whether it's in us or a frog or zebrafish or something, cells and multicellular organisms have to be held together. So you need proteins to hold things together. Cells are phospholipid bilayers, so we'll talk about phospholipid bilayers later on, but I think you guys know membranes are weak. Phospholipid bilayers aren't very strong. They're just sort of these, they're held together by sort of hydrophobic interactions and things. So cells, the cell strength comes from the proteins that are present in cells, including the cytoskeleton and cell adhesion molecules. So we'll be talking a lot about epidermis. So here's an example of skin up here. So skin contains epidermal cells. Epidermal cells have a phospholipid bilayer membrane, but they also have proteins holding things together. So you have many cell adhesion proteins and cytoskeletal proteins that we'll talk about over the next 10 lectures that are basically designed to hold things together, provide structure and support to cells. Especially your skin, you can imagine, you want your skin to be resilient. If you pull on your skin, you don't want it to rip off. So part of the role of cytoskeleton and cell adhesion is to hold things in place. Interestingly though, while you want to hold things in place, sometimes you want things to move. So you can have movement within a cell. So you've already talked about anterograde and retrograde transport, and we'll talk more about anterograde and retrograde transport. You have vesicles going up from the ER to the Golgi up to the cell surface and then coming down as well. So things are moving within the cell. Often you also have cells that are moving. Cells migrate. Cell migration is common in embryos. There's a lot of cell movement in embryogenesis. Parts of cells are moving, nerve endings are growing and moving. In your body, there's immune cells that have to move around and go fight bacteria, etc. So cell movement is also necessary. Movement is necessary. And interestingly, the same proteins that are responsible for holding things in place, these cell adhesion molecules and these cytoskeletal molecules, are also responsible for moving things. So cell adhesion is important in holding things together, but cell adhesion is also important for allowing things to move, likewise for the cytoskeleton, which is inside the cell. It's important for holding things together, but also for helping things move. Okay, so that's all sort of intro. This is still an intro slide. This is an overview slide that just to make a few points. So to look at the cytoskeleton and cell adhesion and something called the extracellular matrix, which is all the proteins that surround the cells, we're going to look at sort of 6 examples over the next bunch of lectures. So we're going to look at sort of protein trafficking, cilium flagella, mitosis, which is made up of karyokinesis and cytokinesis, muscle contraction, cell adhesion, cell migration, and something called extravasation. So those processes are all sort of represented in pictures here. So you don't need to memorize any of this yet because we're going to go through each one of these, multiple lectures. One of the points I'm trying to make here is when you look at sort of images and art like this, appreciate that it's not the whole story. Appreciate that a lot of these things are sort of artistic representation or using antibodies or other sort of imaging to show you something, and they're trying to show you the basics of what they're trying to show you. So in a little diagram here, these cells that are showing these different cell adhesion molecules, is this what it looks like? Of course not. This is some sort of artistic representation of sort of the simplicity of how these molecules can be envisioned. It doesn't mean that the molecules are green and pink. It doesn't mean the shapes are like this. Okay, so when you kind of look at art, don't get fooled, because a lot of times in the textbook things look a certain way and they may change colors or they may change the shapes of things. It's just an artist trying to draw things differently. You know, certain artists may look at sort of the secretory pathway in a certain way, and likewise when you kind of look at sort of cell movement, it can be drawn in different ways. This is just sort of artistic representation of how things look. There's way more going on than what the artist is showing you. Likewise, when you look at fluorescent images, those fluorescent images are of that specific molecule. There's many more things going on, whether you're looking at sort of metaphase here or migrating cell. There's a whole bunch of other things in addition to what's being shown there. So appreciate the complexity of things. Okay? So let's start off with an overview, and while you will have to memorize everything on this slide, we'll go through all of this stuff in other slides, or we're going to talk about things independently. So the cytoskeleton, which is basically these proteins that are found inside of cells that help structure cells, is made up of 3 types of proteins, 3 families of proteins. So there's actin that makes up microfilaments, a protein called tubulin that makes up microtubules, and a protein called intermediate filaments that makes up intermediate filaments. Okay, so we'll talk several lectures on each of these, the least on intermediate filaments. So these 3 are responsible for holding things in place, but also for moving things. So the actin microfilaments are the smallest. They have a diameter of 7 to 9 nanometers. The the tubulin makes up something called the microtubule. Microtubules are the largest. They're a diameter of about 25 nanometers. And the intermediate filaments, as the name suggests, are intermediate. They're in the middle at about 10 nanometers. So we'll be going through each one of these, and a reminder, these are all found inside the cell. So almost all cells have an actin cytoskeleton, a microtubule cytoskeleton, and an intermediate filament cytoskeleton. So you can see here it's the same cell imaged in different ways, showing you these cells have these different components in them. But just to remind you, when you're looking at actin or tubulin or intermediate filaments, there's still lots of other stuff in the cell. I want you to appreciate that in the cell cytoplasm, there is no empty space, right? Molecules are 3-dimensional and they take up volume. We know what a lot of molecules look like, so here, these are some sort of 3D representations of different types of molecules that are sort of showing you the size of things. So you have lipid bilayers, you have RNA and DNA, and this is an immunoglobulin molecule, etc. So these molecules take up space. So it's important to appreciate when you are looking inside a cell that there is no empty space in a cell, right? Water is a molecule. Water can be drawn sort of in this sort of form. If nothing else, there is liquid inside a molecule. So if you see a little drawing like this, which is kind of showing you the microtubule cytoskeleton, right? You can see a nucleus, you have a centrosome here, and these green lines are the microtubules. And it's fine, but that's an artistic representation of sort of a simple cell trying to show you what microtubules are like. When you look at this though, there is no empty space present around there, right? In reality, it looks more like this, right? So we have a microtubule, and in the rest of the cytoplasm, there's all kinds of other molecules in there. You have microtubules, you could have actin microfilaments in there, and a whole bunch of other stuff in the cytoplasm. So for simplicity, sometimes you kind of focus on one thing, but always remember there's tons of other stuff in there that makes it complex. The top right represents a red blood cell, so this is the membrane of the red blood cell. Inside the red blood cell is hemoglobin. Outside the red blood cell is the rest of the serum in the blood, your blood. Your blood is not empty, right? There's always, your whole body is full of volume. There's always something there. Okay, so I'm going to start off by showing you a movie, which will probably take the rest of the lecture time. It's called Inner Life of the Cell. It's too big to upload. You might have seen this before. There are many versions of it online. Some of them are narrated, some of them are shorter, some of them are longer. I'm going to talk over this a little bit and kind of explain things. You're going to watch it, and it's going to kind of look cool. And then at the end of my 12 lectures, you will understand almost everything that is going on in this video, if this works. All right, so this one has strange music on it, I think. Okay, so these are leukocytes floating down your bloodstream. So they're about to undergo a process called extravasation. So they're going to go and fight an infection, and they are going to undergo different types of cell adhesion, and we'll talk about cell adhesion. So this is a leukocyte on your blood vessel, on your endothelial cell, and it's undergoing different sort of interactions. And eventually the point is that this leukocyte is going to extravasate. It's going to leave and go fight the bacteria. But for it to do that, there has to be a whole bunch of signaling cascades triggered. And this is trying to show you all the different signaling cascades that are going to be triggered to allow this cell to extravasate. Right? So there's lots of cool stuff in here. You can see the lipid membrane here, lipid rafts, which we may talk about, but you can see lipids are fluid. You can see that the phospholipids in the bilayer are moving around. You can see that phospholipids are different. There's different phospholipids in one half of the bilayer versus the other half of the bilayer. The two leaflets are different. If you add cholesterol, the fluidity is different, and you often have different types of receptors and things inside lipid rafts. You have all kinds of different interactions going on at the cell surface. And Dr. Cumming will come back later on to talk about a bunch of different receptors and signaling molecules. So this was looking inside the cell, looking up at the plasma membrane, and you have actin and a protein called spectrin supporting things. That's a G-coupled protein receptor, which we may talk about later, and this is talking about different ways to link things into membranes. So this is acylation or prenylation, where you can link proteins to a membrane. So again, you're supporting the plasma membrane. If the plasma membrane is weak, so you need spectrin, which works together with actin to support the membrane. You have to support the membranes. So this is actin. Actin can form bundles, so these are actin bundles. And as you get further into the cell, actin can form networks. So here, actin is forming networks, more like a meshwork of things. As you get further into the cytoplasm, actin can polymerize. So here you can see these actin monomers kind of flying in magically, and when it depolymerizes, it flies away magically. Is that how actin moves? Is there nothing else there? Of course not. This is all full of other stuff. So the way things move isn't as explained here, but things do move around, things do polymerize, depolymerize, things are cut. Just appreciate it's much more complex than this. So that was actin. This is tubulin, this is microtubules polymerized. Again, does it look like this with these things magically being vacuumed in there? No, they have to be brought there, and it's much more sort of a complex process. So that was polymerization, this is depolymerization of microtubules. This is microtubules being used for antegrade transport. So this is a protein called kinesin. We'll learn all about kinesin. Kinesin moves to the plus end of the microtubule, so it's going towards the cell membrane. Microtubules come from microtubule organizing centers, so we'll learn about those. Here's the mitochondria moving along the microtubules. Microtubules come from a microtubular organizing center, the centrosome. You've probably heard about it. We'll talk a lot about it. Inside the centrosome is centrioles. So once signals get to the nucleus makes RNA, and that RNA is going to be translated. So as RNA is translated, you make a protein, right? So you have ribosomes coming on, and eventually you're going to make a protein. What happens to that protein? That protein can stay in the cytoplasm, or the protein can be targeted and moved somewhere else. So in some cases, it's going to be moving. For example, I think this one moves towards a mitochondria. It looks like this thing has a jetpack on it, and it's taking to the mitochondria. It's not just floating through nothing. There's much other things going there. You can also make secretory proteins. So those secretory proteins are going to be taken to a translocon, and they're going to go into the ER. Right, so if you want something secreted, it's got to go into the ER. You can also make transmembrane proteins. We'll talk about how you get transmembrane proteins. From the ER, if you're going to process it towards the outside, it's going to move towards the Golgi. How is it going to move towards the Golgi? Again, our little friend kinesin taking things towards the plus end. So from the ER, it goes into the Golgi. It's going to go through all the different stacks. It's going to get processed. eventually from the Golgi, it's going to keep on going up towards the cell surface. And once it gets to the cell surface, there's going to be exocytosis, my apologies. And with exocytosis, it's going to provide molecules that will then allow for the extravasation of this leukocyte. So now the leukocyte can bind to things, and the leukocyte can leave the bloodstream, and the leukocyte can go fight infection. This is going to happen at some point. Again, I'm just reminding you some of the cell-signaling molecules up there. Eventually, you're going to get to cell adhesion, but you get the idea. And people are eager to leave. Almost there. So this is integrin, which is going to bind to the leukocyte, and then the leukocyte will slow down and extravasate. And it'll be exciting. And then the leukocyte goes off and fights infection outside of the bloodstream. All right, and with that, we'll stop. Thanks for your patience. I didn't do it, all right? See you Wednesday. I mean, in principle, we're in short blood work.
Okay. I just want to so much.
But certainly, I don't do it on while you're traveling. Yeah. I don't know. I don't know. I'll see how it goes. I'm sure lots of really Well, I think because there's say you have the material, so you're ready to present questions. Yeah, so we'll have to sit down and Rob and I and figure out the material that's appropriate to you that is in the picture. So one part of it is will be much more.
拿进来啊！